Welcome everyone for the ELSC seminar series by Zoom. Today we are honored to host Professor Ilana Witten from Princeton University. Ilana did her undergraduate in physics in Princeton University and then her PhD and postdoc in Stanford. She received numerous prizes and awards, among them the Daniel Friedman Prize for Exceptional Basic Research and also the McKnight Scholar Award. And her work focuses mainly on neuronal circuits for reward learning and decision making. So let's welcome Ilana. And Ilana, the screen is all yours.
Let me just share my screen. Okay, great. So I've been told I can expect questions and interruptions throughout, which is fine. So look forward to hearing your questions and sharing some of our work with you. So today's talk, I'll tell you about some of our recent work studying the dopamine system. And as a little bit of background, the textbook view of the dopamine system is that they provide a homogeneous reward prediction error to the brain. So what does this mean? So this is some very classic work from Wolfram Schultz showing the responses here to unexpected rewards drives a nice dopamine phasic response, and also unexpected stimuli that predict reward. Whereas when reward is predicted, in other words, when reward is expected, there's no response. And now I'm giving a talk. And so the idea is that this signal, the reason it's been of such great interest is the signal is thought to be really critical for reinforcement learning. In other words, learning to do actions that lead to reward. And there's really a huge amount of evidence for this, that this signal supports reinforcement learning in the brain. Some of this evidence comes from synaptic plasticity. If you activate dopamine neurons or if you release dopamine, that can lead to strengthening of synapses in regions like the striatum with the neurons that are coactive. And then there's also a lot of behavioral work with optogenetics that activating dopamine neurons is necessary and sufficient for driving reinforcement learning, for getting animals to do an action in order to get that stimulation. And so this has led to, for really many decades now, a classic view of how the dopamine system mediates reinforcement learning, which I'm illustrating with this simple schematic, which is that there are neurons, let's say in cortex, that encode information about the animal's state, the state of the world or the state of the animal. And these neurons converge onto neurons in the striatum. And these neurons in the striatum, their job is to predict the future reward associated with that state or the value. And the way they do that, the way they predict how much reward will come from some state in the world is that they have this dopamine reward prediction error signal that modifies the synaptic weights of neurons in cortex that are coactive with the dopamine. And that modification allows the strengthening of weights such that you actually get striatal neurons that predict value or predict future reward, because again, the dopamine neurons encode the error in the prediction of reward. And so if you use that error signal to train the weights, to modify the weights, you end up with striatal neurons that predict future reward. And that's very useful to do actions that will lead to the most reward. And that's why this sort of model has been so compelling. And in parallel with these ideas that are very compelling about this neural implementation, the same teaching signal is used in artificial intelligence to create very powerful artificial agents that can learn very sophisticated tasks as well. So this is a very compelling view. But things that have led to this picture led to some complication and confusion is the growing appreciation that dopamine neurons don't only respond to signals that look like reward prediction error. There's also evidence about dopamine neurons that encode choices or actions, distance to a spatial goal, locomotion signals, the accuracy of a decision, and also things like aversion or salience. So it's a little bit hard to think about how do we combine the ideas that dopamine neurons encode a variety of things, with ideas that dopamine neurons are a teaching signal, a reward prediction error signal that sets weights across the brain so that neurons can predict value. And so as a first step to trying to think about this question, we wanted to understand how the responses to these diverse features are organized within and across dopamine neurons. In other words, you could imagine one situation with specialization. You have some neurons that do encode reward prediction error, and those are the ones that are involved in setting the weights and learning which actions lead to reward. Whereas there are other neurons that encode other things like movement or decision accuracy, and they just have they're just different dopamine neurons with different functions. So there's just specialization. And just because a neuron releases dopamine doesn't mean it encodes reward prediction error, that's just a subset. Whereas, of course, the other possibility is multiplexing. It's the same neurons that encode this range of features. And this would provide a lot more of a challenge for trying to understand how you get the dopamine system to mediate reinforcement learning. So why don't we know the answer to this question? And or why didn't we know the answer to this question? That's basically because you need basically a task that engages multiple features that drive dopamine neuron responses. And you also have to record at the single cell level from a large population of neurons to understand how across the population there's representation of these diverse features. And so what we did for this, and I should say this was actually Ben Engelhard, who's a graduate from your program. What we did is record or image dopamine neurons during a task in virtual reality, this accumulation of evidence task. So the idea about this task is that mice are running in a virtual reality environment, and they're cues to either the left or the right. And their job is to turn to the side with more cues in order to get reward. So this has a motor component, a decision component, a reward prediction component. And so it has a lot more aspects than the typical Pavlovian conditioning task that was originally used to sort of confirm the concept that dopamine neurons encode reward prediction error. And this is what, just to give you another sense of this task, this is what the environment looks like from the mouse's perspective. So you see there are more cues on the left. So if it accumulates that evidence correctly, it will go left on that trial. And oh, and again, this is work from Ben Engelhard, who graduated from the program at ELSC a few years ago and is starting a position at Technion soon. So so this is what his approach was, which was to implant a grid lens above the VTA and then to image neurons, the dopamine neurons with GCamp, and then with 2-photon imaging. And this was really exciting because this allowed him to record from 15 to 30 neurons at a time, which was much different from the normal yield from dopamine recordings, and allowed him to really get an idea of how the whole population responded during this task. This is the activity under the field of view, and you can see that there are basically, just by looking at this, you can see that there's some moments where the dopamine population is more active than others, and you can see that there's some times where there also seem to be more synchronous than other moments. And so what did he find? What he found is a huge diversity of responses during this task, very different from this image of a homogeneous reward prediction error. He found that different subsets of neurons encoded basically most of the variables that he could measure during this task. So one of these things is position, which is basically the position along the central stem of the maze, where he saw ramps, where many of these neurons had upward ramps, but also some neurons had downward ramps. And this is actually something that had been seen during voltammetry with average recording of dopamine concentration in the striatum, but hadn't been reported at the single cell level. He also saw very diverse tuning for kinematic variables like speed and acceleration and view angle. So here are different neurons with different tuning to these variables. Then the thing that dopamine neurons are most famous for responding to are these cues that predict reward. And so in this environment, there are these cues that tell it which side to get reward ultimately. And he did indeed find a subset of neurons, a little less than a third, responded to cues, but they had very diverse cue responses. They didn't respond similarly to, for example, cues contralateral versus ipsilateral to the recording site, and also different neurons at different latencies in their, or different durations in terms of their response. So there were some, only a subset of neurons responded to cues, and they tended to do so in specialized ways. One of the most interesting signals he saw were neural correlates of what we called accuracy. Whether or not the trial was ultimately going to be correct was also represented by a subset of dopamine neurons, and also whether the previous trial had been correct, which is something that's been classically associated with reward prediction error. And really in contrast to this huge diversity during that central stem of the maze during the cue period where all of these things were quantified, during the outcome period, this looked much more like the classic view of dopamine neurons. Basically, all of the neurons did, or almost all of the neurons responded to reward, and they did so in a uniform way in that they responded more on rewarded than unrewarded trials. So basically, the take-home message is really a huge diversity of responses, but then at the same time, a huge diversity of responses during that central stem of the maze and tuning to very specific variables, but then at the same time, this homogeneity during the reward period. So before we start thinking about what that might mean, I want to tell you a little bit more about how the responses to these variables are organized across the population. And so here, I'm not going to go into the details, but what Ben basically did is made an encoding model to describe for each neuron how much does kinematic variables or position or previous reward or cues, the things I showed you in the last, how much do they contribute to the overall response? What's the relative contribution? And what he found, and it's basically similar to the more raw data I showed you before, is that almost all of the neurons had a strong contribution from reward, but then there was specialization in the cue period in terms of which variables were represented. So there was basically one cluster that responded more to kinematic variables, another that responded more to position, and so on. But all of these clusters had this multiplexing of their reward response.
Question?
So again, yeah?
So how much overlap was there between cells encoding position and other variables during the task?
So basically, like how many of these things are also, so this is like position is this cluster, and basically all the position neurons also respond, well, not all, but the vast majority also responded to reward, but you're saying how many also responded to kinematics, previous reward.
Yeah.
So basically the most, we looked at how many, I don't remember off the top of my head for the position cluster in specifically, but on average, a neuron responded to the most commonly a neuron responded to 2 variables during the cue period, and then the second most common thing was 1 variable during the cue period. So the vast majority of neurons responded to either 1 or 2 variables, were significantly modulated by either 1 or 2 variables, meaning that these neurons that responded to position, like some of them also significantly responded to kinematics, and also some of them also significantly responded to previous reward. Does that answer your question?
Yeah, thanks.
Thanks for the question. Okay, so
another question.
Yeah.
How come you exhausted all the population of neurons? So all neurons respond to one of those variables. I'm not talking about the reward response.
No, this is a plot of the neurons that responded to at least 1 variable.
I see, but how many didn't?
How many responded to 0 variables? Like you're saying
no variables that you measured.
Right. So you're saying of the neurons of these 246 neurons that responded to reward, how many didn't respond to any of these other variables?
Yes.
I don't actually remember the number off my head. It was like the majority responded. It wasn't all of them. I don't remember the exact. It was the majority, but it wasn't all of them. 80%, I think 246 out of 300 is about 80% responded to reward. And then that other plot was of the ones that responded to reward, the ones that also respond to at least one of these. And it was definitely the majority. I don't remember how many ended up getting excluded from that plot. And again, of these neurons, most of them actually even responded to 2. So they didn't respond to all 5, but 2 was the most common, and then 1 was the second most common. But there's some that responded to 0. I just don't remember the fraction off the top of my head. Oh yeah, so basically, just in the interest of time, I'm not showing you these plots, but you might wonder, how is this heterogeneity? How does it map onto the VTA? And so the one thing that Ben did was reconstruct the location of these neurons within the VTA. And there was a noisy spatial organization, meaning there was a significant relationship between some of these clusters and where they are in the VTA. For example, accuracy neurons tended to be more medial, whereas kinematic neurons tended to be more lateral. And then another thing I'm just not showing you in the interest of time is that if you move away from this evidence accumulation task where so many things are going on, the animal's running and it's turning and it's accumulating evidence and has had recent rewards and may have reward in the future. And if you go to the simple Pavlovian conditioning task where there's just a cue that predicts reward, then you basically don't have heterogeneity anymore. You have this low dimensional task, and then you have this very low dimensional dopamine signal where there's just, it looks exactly actually like the data from Wolfram Schultz from the monkeys that I showed at my first slide. Dopamine neurons just respond to the cue that predicted reward. All of them do. The same neurons actually also we tested in that setting, and also to unexpected rewards. So one thing that is striking, again, is that instead of all this heterogeneity for these task variables during the cue period, the reward response is pretty homogeneous. But I talked to you about dopamine neurons as a reward prediction error. Here, I'm just quantifying reward response. So a question is this reward response actually a reward prediction error? Is it signaling the difference between how much reward the animal got and how much was expected, or is it simply just a reward response? And luckily, there are at least 2 ways to quantify this in this data set. Basically, what we need to do is compare reward responses on trials when the animal expected reward, had a high expectation of reward, versus trials where the animal had a low expectation of reward. And so the idea here is this is the trial difficulty idea. So there are some trials that are easy. There are a lot of cues on one side and no cues on the other. And so it should be, the animal should know, like, I'm definitely going to get reward. Whereas there's some trials that we call hard trials. There's a similar number of cues on the left and the right. And it's actually just, if they're equal number, it's just 50-50 whether the animal will get reward. So they should have a different internal expectation of reward in these 2 cases. And then the other dimension of reward prediction error that can easily be gleaned from this task is based on reward history. Animals tend to expect reward. This has been shown many times that animals tend to expect reward if they've had it recently, whereas if they haven't had it, they don't expect reward. And this is even true in tasks like this, where the outcome of one trial actually doesn't carry information about the outcome on the other. Each trial is independent. This is still just a feature of animal trial and error learning and animal behavior. So basically, if the previous trial was rewarded, they would have a higher expectation of reward than if the previous trial was an error trial. And so these reward responses on average across the population are modulated by reward expectations, so that you can think of both of these, through both of these measures, you can conclude the reward response really is a reward prediction error. If you look at trial difficulty, there is a bigger response on the hard trials when reward is more unexpected than on the easy trials on average. And also, if you look at previous reward, the same idea. There's a bigger response if the previous trial was an error, so reward expectation would be lower than if the previous trial was rewarded. So what, for this first part of the talk, what I've told you so far is basically there are really widespread and homogeneous reward responses, which we really think of reward prediction error responses. But this is what's surprising, is that this is multiplex on the single cell level with very heterogeneous and specialized representation of task variables.
Question?
Yeah.
So, but the the dependence on the prediction part, what you show is kind of average, but maybe there is also clustering in this.
Yeah, because it looks very, I mean, these are two different dimensions of word prediction error. Like, you know, in a Pavlovian conditioning task, there's really only one dimension. And as one thing we've like really realized from this work is as the task gets more complex, animals could use many different dimensions, at least two in this case, to predict reward. But as you're pointing out, there seems to be a lot of, on average, things are reward prediction error, but there is a lot of heterogeneity. Whether that's because this is only, you know, this is only two possible ways to predict reward, and there might be even additional ways, or because not all of the neurons are actually predicting reward is hard, very hard to rule out. But your question was a little different. It was, is there clustering? I mean, the only thing we did is look, your question is there clustering of the reward prediction error response?
Yeah, so maybe that some of the neurons just are reward response, period. And some of them are more selective to an error, prediction error, and other ones are more selective to the history. So,
right.
so just to say that there is a uniform reward, you know.
No, you're right. All we're saying, all we can really say from this is there's a uniform reward response, and then on average, that reward response is modulated.
I know, but
yeah. So the two, I mean, we've looked for structure in this, and to be honest, have not seen a lot of structure. So we don't know if it's because we don't know exactly what each animal or what each neuron is using to predict reward, or just because there's no structure. Like, for example, we just plotted on each neuron these two dimensions of reward prediction error against each other, and there's no obvious structure. And then we also, for all of our clusters that were defined by the Q period, I think we put this in our paper actually, just how much each of these, you know, we thought maybe one cluster would be modulated, like the neurons that were modulated by previous reward during the Q period, maybe they were the ones that had this type of reward prediction error and not this type. But it wasn't, I mean, there might be some structure and maybe the ends become too small, but it wasn't obvious. It wasn't super clear. So we don't know if that means there is no structure, or we don't have enough data, or that we're not knowing exactly how each animal is predicting reward. It's a good point though. At this point, all we can say is on average, the reward response is modulated by expectation in at least two different ways. And whether there's more structure in that, like, we just don't have an answer because we haven't found it, but that doesn't mean it's not there, I guess is what I'm trying to say. Okay, so so basically, oh, and then I'm just repeating what I just said 4 times, which is that on average, these reward responses are modulated by reward expectation. So, so basically, what does this mean for us? So there's 2 things that I'm going to tell you about for the rest of this talk. The first
question is also in the chat on the first half.
I'm not looking at the chat, but can someone just read the question if they want me to answer it?
I can read it. It says, isn't it surprising that the Q cluster is so small, considering Schultz's results on strong DA response to CS? Would we expect to see many more neurons responding to deterministic cues instead of these accumulating cues?
Yeah, so we did run yeah, that's a good question. And I probably said it too quickly. So we ran mice through like the Schultzian, like 100% predictable cue to reward. So, you know, these cues are only somewhat predictive of reward, right? They're, you know, some cues are more predictive than others, and no one cue tells you with 100% certainty you will be getting reward. Whereas the Schultz situation is Pavlovian conditioning, and there a cue predicts reward with 100% probability, and at least in the most basic paradigm. And so when we do that, suddenly all of the dopamine neurons respond to the cue. So we do think you're right. It has to do with how predictive the cues are of reward. All right, so so for the rest of this talk, I'm going to tell you about 2 things we've been thinking about more recently. One is a question we've gotten from a lot of people who record spikes rather than calcium, from both from dopamine neurons and from other neurons in the brain, which is, I think, an important question, which is some of these unexpected responses that you see during this task nothing to do with the task, and more because you're measuring calcium, and how can you compare that to everyone else who's measured spikes? So what I'll tell you about next is a little bit on work we've been doing recently, thinking about the relationship between calcium and spikes in dopamine neurons. So it's a little bit of a detour, but I think it's important. And then the last part, I'll tell you about some new work we've been thinking about is now that we see this heterogeneity at the single cell level, how can we, and we see it's actually the same neurons that have reward prediction error that encode these task variables, if and how can we reconcile this with reinforcement learning models? And the reason I think, again, this is important is if we had found and said that it's the same, if we had found and said it's different neurons that encode reward prediction error versus encode movement and these other things, there'd be no need to reconcile it. You just make a hypothesis that it's like different neurons have different functions. But once it becomes the same neurons, I think it becomes really important to start to think about how you can layer this data into the idea that of reinforcement learning, where dopamine neurons are important in learning which actions or stimuli predict reward. So that'll be the rest of my talk, these two parts. Okay, so the calcium part. So the question we've gotten a lot is, can some of this have to do with measuring calcium and not spikes? And one particular version of this question has to do with these ramps we see, these spatial ramps with position we see in the calcium response. In other words, could spatial ramps be a consequence of calcium integrating a signal that's not ramping? Basically, calcium's slow, it's an integral, or do we think the firing rates are themselves ramping? And so that just made us take a step back and realize that people haven't really validated spike inference in really anywhere outside of cortex, basically, and definitely not in dopamine neurons. And even in cortex, there's been some recent controversy about how relatable is calcium to spikes. So what Wes and Fleming in the lab did is image calcium and record spikes in slices as a first step. And what you can see just looking at this is the spikes in the calcium in this preparation look very, very closely related. And so we wanted to sort of take that a step further to see exactly how well could we relate them. And so we took advantage of a model for calcium dynamics that had been proposed by Liam Paninsky's lab, which is that the observed, Y is the observed fluorescence, and it's based on the underlying calcium plus some noise. And then the other part of it is that the calcium is decaying with some decay rate, that's like the decay of the calcium, unless there's a spike. And when there's a spike, the calcium pops up. So here there's a spike, so the calcium pops up. And the black here is the observed fluorescence. So this model leads naturally to a straightforward optimization problem, which is basically that the fit of the estimated calcium to GCamp plus the number of estimated spikes with some penalty lambda, so this is the second free parameter, it has to be minimized to give you the best, to get you the best estimate of when those spikes happen. And this is something that has been an approximation to this problem is what Liam Paninsky's lab used. And we've actually been working with my sister who's a statistician at UW and who realized that this could also, you don't really need to approximate this problem. There's some methods from dynamic programming that actually allow you to exactly solve it with only this one free parameter plus the decay rate. And the reason we're excited about this approach is therefore, like what makes spike inference hard is that there's a lot of free parameters and it's very arbitrary how exactly you choose them and your choices can affect your result. But with this approach, you only have two free parameters and you can observe them both. So we don't fit the free parameters. The first is the decay constant of calcium in dopamine neurons, which we measured in vitro, just fitting the decay during periods where there weren't spikes. And then the other is that, as I mentioned, that penalty on the spikes. And that just basically will determine how many spikes you infer total. And there's been a lot of recordings from dopamine neurons. So we know how many spikes we want. We want like a firing rate of about 5 hertz. So we basically don't do any fitting. We just set those two parameters based on like our knowledge of the biology and then use that to fit the spike, to find the times for these spikes, the inferred spikes. And that works pretty well. This is with the in vitro data. You can see a nice match to the estimated and observed spikes in some typical cells. And the green is estimated and the black is observed here. Here's another example neuron here. And so this is in vitro. We then took the same approach on the in vivo data. And so this is just unexpected reward, just to show you some example data. These are 5 trials of unexpected reward GCamp, and this is just where the spike inference thinks the spikes happen. So it estimates both some tonic activity, just like dopamine neurons are supposed to have, and some phasic activity. And this is showing you what an average of 65 inferred spikes from 65 neurons based on calcium, so inferred spikes from 65 neurons to unexpected reward. And then this is EFIS data to unexpected reward from now Uchita's lab, which is a dopamine electrophysiology lab. And so we're pretty happy with this, because again, without like fitting the free parameters, we get like a tonic level of activity that looks pretty similar to the dopamine neurons. And then the phasic activity also goes up to 30 hertz, which is also pretty similar. And the other thing we think it looks promising in terms of just how relatable calcium is to spikes is we get another famous thing about dopamine neurons is the pause in activity when they expect a reward, but it's omitted. And so here's a conditioned stimulus that predicted reward was presented at -2 seconds, and then at 0, that reward never came. And so we do see a pause, and we actually see this in a similar, in the inferred spikes in a similar proportion of neurons as was reported with electrophysiology.
Can I ask a question?
Yeah.
So in the previous slide, here, so it seems like in the inferred spikes, the firing rate does not go back to baseline, while in the electrophysiology recorded from neurons, it does.
Yeah.
So yeah, can you explain that?
I don't know. It's definitely not true about every neuron, but there were definitely neurons in our population that had that. So yeah, the firing rate there. So that's a good point. Like, and whether, so I mean, another difference is the second peak in the EFIS. And we don't also know, so I guess we don't know, this is just what the data that we have shows, but the second peak in the EFIS might be something we can't pick up, but it's actually possible. If I understand correctly, there might be people who know this better than me. You only see this second peak, or you only see this in certain situations in dopamine neurons. And in particular, they have a lot of different reward sizes, and we only had one reward size. And so this reward size was better than the average reward size. So that second peak is like, it takes a while to get that, like, oh, this response was better than I would get on average. And we were actually missing, like, we just had, this is data we already had where we didn't have the same kind of collection of reward sizes. So that might be why. So, I mean, yeah, so this is, so I guess I don't want to say they are necessarily identical, although you also have to say, realize these are also different neurons from different mice. So I don't know if we fit in a slightly different paradigm, but again, we didn't even know if you'd be able to recover tonic firing from these types of models, and also if the phasic firing would be able to.
But it seems that return to baseline is a much more basic criteria for success of the model than the details of the peak.
Yeah, I guess I had not thought too much about the return to baseline, since we haven't been thinking so much about that, but we should look at how many neurons, like what neurons are contributing to that in the average. That's a good question we hadn't, to be honest, hadn't thought about yet. Okay, so the question is basically like, if we apply this to the fluorescence, do we get ramps that look similar, like to how do the estimated spikes look compared to the fluorescence? And so basically, according to this model, of the neurons that ramp tend to have a change in firing rate of a few hertz, about like, around 4 to 8 hertz in their baseline firing rate. And there's actually a new paper on bioRxiv measuring electrophysiology as animals traverse a T maze. And they're also seeing changes of about this magnitude. Ramps lead to changes in tonic firing of about a few hertz. So that we find promising as well. And similarly, we also see that the decrease in some fluorescence can be end up looking like a downward ramp in firing rate. So basically our interpretation is that the calcium and the spikes seem pretty relatable to each other in dopamine neurons. So just to summarize what I've told you so far, for the first part of the talk was that there's in vivo calcium imaging reveals a lot of heterogeneity during decision making, specifically heterogeneity to task variables during the Q period and as the animals in the central stem of the maze, and then more homogeneity during the reward period. And then we think that the spikes can be inferred rather accurately from the calcium imaging data. And so that suggests, at least to us, that that our data isn't, our interpretations don't come from measuring something that's very different from the spike.
But question, but the spikes that the firing rate are kind of average, trial average. So it's still, even if it is spikes, it still can be the case, like Jonathan Pillow's argument in other cases, that even if you look at spikes, that individual trials will have kind of more underlying jumps which are smeared by trial average.
So you're saying,
Jonathan Pillow's conundrum, this is spikes, it's not calcium.
What?
Like Jonathan Pillow's argument.
Right, but that's a single trial argument. Just to be clear, everything I've shown you is trial averaged. Everything I've shown you today. I haven't shown you any, I've made no single trial argument.
Okay, so it can still be that single neuron is quiet and turning on and at different times in different trials.
A single neuron is turning on. So you're saying that maybe
different trials are just different times when the neuron fires.
Yeah, that like a neuron that is one way of saying it, like a neuron might ramp on some trials, but not on other trials.
No, doesn't ramp at all. Just switch on at a given time in the trial.
Oh, I see. Okay, yeah. I mean, you're saying, okay. Oh, you're talking literally about Jonathan Pillow's story that in LIP.
Literally, yeah.
I thought, I didn't know you were literally talking, and with respect to ramp, I thought you were talking more about the spike inference. You're saying literally, okay, you're saying that like Jonathan Pillow says like LIP neurons on a trial by trial basis aren't ramping, they're like ramping on average. Yeah, I mean, we definitely haven't applied his analysis to our data. I'll say, I wish I actually were showing you individual trials. I don't have that in this PowerPoint. I don't think, I mean, I don't know what the LIP data looks like. That's not what individual trials look like to me, but like, yeah, it could definitely, it's not that he says they don't, yeah, you're right. He says they just step on different trials. Yeah, I definitely, it definitely doesn't look like a phasic burst in dopamine neurons, but I suppose a step that gets blurred out across trials. Yeah, we definitely can't rule out, and we haven't run his analysis on this data. Yeah, we haven't like explored that idea at all. I didn't even understand your question because that's how far we were from exploring it. But yeah, I mean, I haven't really thought about it, but yeah, maybe the idea that the ramp could be the average of steps is definitely not something we've explored. It's not what the data looks like by eyeballing, but of course, that's not what the LIP data look like to people by eyeballing either, and we haven't done any analysis. So yeah, it's a good question. Ultimately, to the downstream neuron, those two won't be very different because if different dopamine neurons were to be stepping at different times, obviously it would still look like a ramp in terms of dopamine concentrations downstream. But it's a good question we haven't thought about like at all. Okay, so then the last thing I just wanted to spend, I don't know what the time looks like. Yeah, we have plenty of time. So I wanted to spend a couple of minutes just talking about work we're starting to do with Nathaniel Daw, who's also at Princeton, thinking about if there are ways to reconcile dopamine neuron heterogeneity with reinforcement learning models. And so what I had mentioned in the beginning is that the really thing that's been appealing about this scalar homogeneous reward prediction error that projects downstream is that it's exactly the signal you want. And actually, it's exactly the signal used in machine learning algorithms to predict future reward. It encodes an error in the prediction of future reward. So if you use that signal to update the synaptic weights of neurons that encode the state of the world, you get let's say, striatal neurons, which is the traditional neural mapping of this model, that encode the value or the predicted future reward of the state or of a particular action. And that could allow you to learn to do the actions that lead to reward, or choose the actions that are more valuable. And so here I'm just saying what I just said in words with equations, which is that the cortex encodes the state, and then the weights of the corticostriatal neurons are updated with this reward prediction error signal multiplied by the presynaptic firing of those cortical neurons. And that will give you value encoding in striatum, how much reward is predicted in the future. Again, assuming the dopamine neurons encode the error in the prediction of reward, which is calculated with this temporal difference error, which is the difference in value in neighboring time points plus a reward input. So this is, again, basically a very, very powerful idea about how learning works, how reinforcement learning works. And it's a little bit, so then the question is there any way to fit our data into this framework? And so one idea we've been thinking about is a generalization of this scalar reward prediction error model to think about a vector of reward prediction errors. So now basically it's actually the same ultimate equations, but instead of having a single value in the striatum, you have a whole vector of values where each cortical neuron that encodes state is projecting to in a more one-to-one way, less, rather than converging onto a single value, you have a more labeled line convergence onto a subset of the value neuron. So this neuron is going to encode the value, calculate the value of this state. And then the reward prediction error within each neuron isn't this is the dopamine layer. It's a vector of neurons where each dopamine neuron still is trying to calculate reward prediction error, but only based on this feature, only this subset of the environment or the internal state. So then if you add all the dopamine neurons, you actually recover the same reward prediction error signal you had in the previous slide. And so you still recover a functioning reinforcement learning network. But on the level of individual dopamine neurons, you see something very heterogeneous that where you'd still get a reward response because all the dopamine neurons get a reward input, but they're calculating basically the prediction error based on different subsets of the environment, different subsets of features. And so we've been really interested in this model, both in its ability to potentially explain our data, but also in making new predictions about how states that predict a reward are encoded in the brain and how that in turn is reflected in the dopamine system. And so as a first pass in thinking about this, Rachel, a graduate student, has been running a simulation of this model. And so this is instead of our mouse doing our virtual reality task, we now have a reinforcement learning agent, a deep learning agent who's running the task. So this mouse actually shouldn't be here because there's no mouse anymore. It's the same task, it's the same software, but instead of a mouse, it's this network. And this network extracts features of the environment with convolutional layers. And then these convolutional layers go to some recurrent layers, some layers that have memory. And then these recurrent layers go to project either to instead of a single value neuron, which is how these types of networks are usually demonstrated, to a whole vector of value neurons that go then to a vector of reward prediction error signals that then together modulate the weights, both the weights that become the value layer and also the weights that become the action layer. This is called the actor-critic network. And so these actions, the agent can either go left, right, forward, and so on. Those then are fed into the virtual reality environment in closed loop to determine what the agent does on the next step. So this model can learn the task. This is just the agent doing it. It's a little jumpy. It actually becomes better with more training. This is before it's reached like plateau performance, but you can see it veers to the side with more cues. And ultimately—
Can I ask a question?
Sure.
So the reward in this case is a vector of the same number? What is the reward?
All the neurons receive a reward. It's just a reward, a scalar reward.
But it's the same reward for every neuron. So why would the different indices of the output vector predict any different number?
Why would the different I don't understand the question. Why would the what output vector? I don't know what it is.
The value prediction vector.
So you're saying where, yeah.
So why would the different nodes predict a different number if they're trained on the same value as each other?
Why would the nodes of what predict a different number of what?
Because the states are different.
To each value neuron, I'm not sure, sorry, I might not understand the question, but there's 64 neurons in this LSTM layer, this like recurrent layer, and each of these neurons predict, like each of these neurons is basically a different value neuron, which then it calculates its own RPE.
But isn't it, sorry, I think I'm misunderstanding. Isn't it trained, isn't the prediction is the reward and there's really only a single reward?
No, the prediction error is basically, it's the temporal difference prediction error. It's like reward plus the difference in the value in neighboring time points.
Okay, thank you.
Sorry, was there another question?
I think because the states are different, but I think functionally, I think I agree with the question. It just, you can collapse everything to one computation and you didn't gain anything. You don't show that you gained by doing this.
So it's mathematically equivalent. It's not a gain, it's to try to explain the data. It's not, it's mathematically.
So what's the point? So what's the point of the simulations if mathematically they're equivalent? So you can just do the usual simulation.
I mean, it's the same simulation. It's not a different simulation. It's just to illustrate this, what dopamine.
Well, okay.
It's not, it's mathematically the same, so it's not, meaning it's not different. It's not a different simulation. It's an illustration of how you can generalize from a scalar to a vector RPE as a way of trying to explain the actual neural data.
But it's the same. It's just the same. It's mathematically is the same. What else one can say if they're mathematically the same?
You can ask if the dopamine neurons that are recorded look like this vector of reward prediction errors.
That's the deep network simulation will not answer this question.
It will answer in the case of the deep network. You're saying because the brain could have a different feature space, will have a different.
No. I just don't understand what the simulation are adding.
The point of the simulation is to extract the features from the environment to then calculate the vector of RPEs. Like, how would you? I mean, how would you do this? Like, basically, it's a very high dimensional environment. What the deep network is achieving is learning the features of the environment, so you can plot out a scalar and a vector of RPEs. So,
okay, so the main point is how they extract different features.
Chaim, maybe we should keep this to the question section.
But yeah, no, it's a good point though. I think you got it. I heard two voices at once, so I might have misheard what you said. But exactly the point, since the scalar and the vector RPE are mathematically equivalent, it's not a different simulation. I'm just illustrating. The point of this is it's a high dimensional environment. What are the features that get extracted, and how do they look in the vector RPE versus the scalar RPE framework? Does that make sense? I didn't hear anything. Okay, so basically the result of the simulation is that basically you get a lot of heterogeneity during the Q period. And this is just one example of that, is if you look at the Q responses during this Q period for the 64 features, and these are the 64 reward prediction errors, you get some neurons tuned to left Qs and some neurons tuned to right Qs, similar to what the actual biology shows, rather than just a single RPE. But in contrast, during the reward period, the reward responses are much more homogeneous, and that not all, but most neurons have a stronger response during reward versus no reward. And I should say that also most of them have modulation by reward expectation, because they are basically the job of the network is to predict the value of the environment. So it basically replicates by design this basic feature of getting a heterogeneous response to the Q period and homogeneous response to the reward period, which was the basic finding, while also being completely consistent with a reinforcement learning framework. And one thing that I think is interesting about this is it leads both to much more detailed predictions about the nature of the heterogeneity you see in the dopamine system, and also how that vector of reward prediction errors is going to reflect the demands of any particular task, and also an animal's internal cognitive state. In other words, how that animal is predicting reward in the environment should be read out through looking at the vector of reward prediction errors in the system. So I think it's still, this is basically demonstrating you can get heterogeneity during the Q period and more homogeneity during the reward period from this model, but it's also just an open question to what extent this can actually explain the details of our data set, the specifics of our data set. So to summarize, the dopamine neurons are pretty heterogeneous during the Q period, homogeneous during the reward period. We don't think it's easy to explain this based on the recording modality. Basically, if you're thinking about calcium instead of spikes, I think that's probably not an easy way to explain this heterogeneity. And maybe a better way might be to think that the standard model is correct, but instead of dopamine being a scalar, it's a vector. And I think that it's an open question to what extent this way of thinking about it can explain the myriad of heterogeneous responses that both our lab, but also many other labs have been starting to observe in the dopamine system. So that is the work I was going to present. So Ben was the lead person on the first study on the 2-photon imaging, and Weston did the spike inference work with Sean Jewell, and Rachel Lee is the grad student working on the vector RPE model. So that is everything I was going to share, and also want to really thank my collaborators and thank you guys for listening.
Thank you, Ilana, for a fascinating talk. So let's switch to questions. Let's start with students. Any questions from the students in the audience?
I have a question.
Yeah.
So do you have any idea as to the functional goal of coding the reward in different heterogeneous way? Maybe this relates to Chaim's question from before, meaning if we eventually get the same reward at the end, the scalar reward, even if you do model the hidden layer as something different, and it matches the empirical data, what would be the functional difference between this and
computational utility? Like, does this have this is probably why Chaim was bothered by this. Like, is there a computational utility, or is it just what the biology is doing? Like, is it just providing the explanation for what's a really like hot topic in the dopamine field, heterogeneity, is just explaining the data, or does it have a computational utility? Well, if it's literally mathematically the same, clearly there's no computational utility. It's computationally identical. So how could there be a utility? Ultimately, I think I'm not sure, because clearly they're only going to be identical if there's complete convergence of the dopamine signal. We know right at the target layer. Otherwise, they're not going to be computationally identical at all. You'll have different like microcircuits that are calculating completely different reward prediction errors and making totally different predictions about value based on only a subset of all value. And you'll have weird interference between them, between different like value calculations on these different like parallel lines. And so my opinion is that, well, I mean, we know that there's parallel architecture in the basal ganglia. We know that there's not total convergence of the dopamine system. And we also know from the work I did and show you, but mentioned briefly, that that the different like RPE, the different dopamine neurons that are heterogeneous are spatially localized. So this is an approximation that that you'll actually recover the original model. It's not correct. Instead, you're going to have these different microcircuits. If this concept of this model is right, you're going to have these different parallel loops that are doing different computations. So it'll be much more parallelized than this model will give you. So it'll give you, whether that's going to benefit the agent or hurt the agent, I think is an open question. But like I definitely don't know. We definitely don't know the answer, but ultimately the idea that they're identical is an oversimplification based on this being wrong. And so once you start to actually have these different parallel loops that ultimately have to compete somewhere for the animal's behavior, and neither is actually going to have the correct value estimate, you're going to have a different situation. And whether that could be useful in some cases, where basically the way I think about it is like, there's a lot of different ways animals can predict reward. So a lot of different, like in a high dimensional environment, there's lots of different dimensions. And so you have these different microcircuits that could be doing that in different ways. And maybe that's useful if you don't want to like rule out any one hypothesis, instead have all the hypotheses going in parallel. So I guess the point is, we don't know. It might just be a problem for the circuit and not a use. But in reality, these two models aren't going to be actually identical. Oops, sorry, I thought I was sharing my screen, but I wasn't. So you didn't know what I was pointing to. Oh my gosh, did my answer make sense?
It made sense. It made perfect sense. But you were showing the whole top, right? Just right now, it didn't show the screen. It's fine.
Oh, but I was like pointing to like things on the screen. Okay.
Okay,
I did share my talk in general. Oh, that's good. Did that answer your question to some extent? I mean, it's an open question. It's a good question.
You did.
Can I ask another question about the data that you showed before, showing that the dopamine neurons are reflecting different features in the task? Do you think that that is a result of those features in other conditions predicting the reward? So the original, we're still having the dopamine neurons predict these or encode these features because they're predicting reward. And this is like perhaps some way for the network to generalize is to to find the features that predict the reward in general and to have representation of those features.
Yeah,
I don't know if that
yeah, no, I definitely, yeah, that's kind of what I was trying to get at. Like, we don't know to the extent with which that's true, but like the idea is even in this task, like even though the animal's velocity doesn't actually predict reward, the animal is moving at different velocities on different trials. And like the neurons that primarily get velocity inputs could be trying to use velocity to predict reward because they're part of the, you know, dopamine vector that gets velocity inputs. So they're trying their best, even though they don't do a great job, they're trying their best. Even though they don't successfully predict reward, they're trying their best to predict reward, and that's the main input they have. And sometimes velocity like correlates with reward, and to some extent it is. If they were literally not moving, they wouldn't get any reward. So I think the answer is yes. That's exactly what we're thinking about. Like there's like in a task like this, the feature space is really complicated. We don't know what it is. The state space is really complicated. Again, one point of that simulation is showing, at least for a deep network, you can know what the feature space is. And then, yeah, and that could be why, like, it's not literally correlates of task variables. It's correlates of like the prediction, the animal's best chance, the neuron's best effort at predicting reward based on that task variable. That's exactly what we're trying to say. Thanks for clarifying.
So just one more clarification. So we would say that that it's using velocity because previously velocity has in the past been predicting reward, and that's
hardwired. Like even like the dopamine system could be hardwired to expect in like sometimes velocity predicts reward. So it could be evolutionarily learned velocity can predict reward. So there are dopamine neurons that get velocity inputs. You know, because it is like lateral VTA. We saw that, which is near substantia nigra, where other people have talked about velocity correlates and dopamine neurons in substantia nigra. So maybe it's hardwired that those lateral VTA neurons get inputs from like motor areas, and because like either through past experiments or through evolution, they know that sometimes velocity predicts reward.
Thank you.
Yeah, thanks. These questions are making me realize this is the first time I presented that last part, so it's showing me I need to unpack it more. So thanks for the questions.
I have a question regarding the first part of the talk. So it's a bit about like you have these neurons essentially representing two features or one feature, like, you know, kinematics, position, etc. Is there any kind of spatial organization of this or any kind of functional organization of the neurons, like different kinds of neurons which do this have different genes or something like this?
Oh, we don't know about genetic anything, but molecular anything, but we do know that they tend to have a spatial organization. Like not all of the clusters are significantly spatially mapped, but the velocity cluster was more lateralized in the lateral VTA close to SNC, and the accuracy cluster was more medial. So yeah, but not all of the clusters. But given they have a spatial organization, I assume there's some genetic wiring there. So I think those are just parts of the VTA, you know, different parts of the VTA have different inputs on average, not completely, but on average. So I just assume the lateral VTA gets more velocity input, and the medial VTA is more connected to prefrontal cortex. So maybe more about like the cognitive state of the animal, whether they're in a state to accurately do a trial or not.
Can I ask something, Ilana?
Yeah.
Hi. So I'm missing something in your diagram, and I'm wondering if it's like completely obvious or it just kind of I didn't get it. But I would expect that the reward should come somewhere in the diagram, and it should probably cause plasticity between these value neurons and the dopamine neurons. Is that part of your model, or is that kind of, or is that assumed?
Reward comes, like the dopamine signals bring information about like reward, like error in the prediction of reward. So the dopamine neurons get a reward input, but also like the difference in value across intervening time points. And together that gives you like reward that's unexpected. So the dopamine neurons encode reward, like unexpected reward. And that's how the weights in the corticostriatal pathway get reweighted to predict the expectation of reward in the future.
Wondering if there's like a assumed plasticity in the output between the value neurons and the dopamine neurons, if this is part of the model.
So between the value, I mean, the way the model is useless. Yeah, maybe.
So look at the diagram. Can you show the diagram?
Sure. I'll try to do a better job then. I don't even know which diagram. Are you talking about this one?
Yeah. So that one, right? So these dopamine neurons, they encode the reward prediction or the reward. It doesn't matter. But at some point, the animal gets a reward, and this should probably affect the dopamine neurons.
Yeah. So you're saying, why did I, like, basically why, like, this is what the equation is for the reward prediction error. It has a reward input, but you're saying just I should have drawn the reward input.
No, I just, if you assume some, if the mapping between the value neurons, the striatum neurons, to the dopamine neurons is plastic, and is that kind of changes as a function of, the activity of the dopamine neurons in a way. Because at some point, right, so in some ways you could interpret your results in the following way. So during the task, the striatum neurons are activated, and they actually compute the prediction, and
right.
they project it to the dopamine neurons, that's your activity that you see.
Yeah.
And the reward comes, it's also activate the neurons independently through the pathway, right?
Yes.
And kind of activate the neurons, and then this is why you see them also active always when
right.
they reward.
Yeah.
And then this could actually cause plasticity between the striatum neurons and dopamine neurons that kind of let them predict better the reward later on when there is no reward.
So basically you're saying this model only has weight changes here, but you're saying there could also be weight changes here.
I think there should be critical weight changes there, because this would what kind of later on make these neurons indicative if there is a reward or not, if the prediction.
Well, I mean, the neurons, this model is sufficient to get a perfect like reward prediction error. Like this sign just means some temporal differencing of the MSN activity. And then this, I didn't drop it, there's a reward input. So this gives you an RPE. So you don't need an extra site of plasticity to get an RPE, but you're correct that there are many other sites of plasticity besides this one. So this model is clearly oversimplified it, but I don't know when and what the plasticity here is exactly for. Like basically all I'm trying to say is you don't need plasticity here for this model, but you're right that there's a lot of evidence there is plasticity there. So I think it's a good question. What does it do?
Can I ask?
Sorry.
Can I ask a question?
Sure.
So thanks for the talk. So early on you had, you were showing that you could tell apart just value or just reward and reward prediction. I was wondering if you could do the same for the other variables. So saying the difference between position and position prediction error and so on. So can you tell the difference between the actual quantities and the errors?
Yeah,
responses? I
mean, that's a really good question. And that's what we need to do to really prove that the vector RP model is right. That was just like, it makes sense in concept, but we have not proved it's correct. And we're trying to work out the trial by trial predictions from the model. And the reason it's tricky is it really depends on exactly how the state is represented in the brain, and also the algorithm the animal is using to update its.
But let's say the Ushida teleport experiment might, the teleport.
But that's not about I'm not sure it would necessarily.
They just jump in the middle of the
Yeah, but
virtual days.
So you're saying if we jumped the animal, I mean, if we jump the animal, we'll get a jump, but that won't tell us that the position. I thought you were asking a harder question. Yeah, I'm sure we could replicate the result. I thought you were asking a harder question, which is if the position correlates are like, how do we know if it's a correlate of position or a correlate of a prediction?
Yes, yes, that's exactly what. No, I was just thinking. No,
Yeah,
but
that's a hard question. But that's what we're working on. We're trying to figure out how specific. That's exactly what we're working on. It is actually a really hard question, but that is exactly what we're working on. Like, are there predictions that really differentiate the model? Like the data is like could be consistent with this vector RP model, but we haven't like proved it is. And so the question, the kind of open question is, are there like trial and error predictions about the position correlates that like clearly differentiate a correlate of position with a correlate, like exactly what you're saying, but it's, and I'm hopeful there are, but we haven't like nailed it yet. Like predictions that clearly differentiate those two concepts. It's a really good question.
So I think we have time for one more question. Chaim, wanted to ask us something?
I would like to ask Didi, can I?
Yes, Hagai, go ahead.
Hagai.
Go ahead.
Hi, Ilana, can you hear me? Good talk, thank you. Ilana, two questions actually. One, if you can make the difference between a VTA neuron projecting to the nucleus accumbens and to the frontal cortex, and do you see different clustering between the two populations? And then if from your point of view, VTA and substantia nigra compacta is a spectrum or two entities?
Oh, okay, yeah. So, okay, unfortunately, we haven't been able to successfully combine retrograde labeling with the cellular resolution imaging. We just don't get, so far in dopamine neurons, we haven't managed to get bright enough retrograde labeling to see the labeling well. But there's supposedly the neurons in, there's supposed to be a spatial mapping between where the neurons are in the VTA and where they project. So the very medial VTA neurons were the only ones that were really tuned to like task accuracy. And those neurons were supposedly either projecting to medial shell or medial PFC, but we don't know that for sure. It's just based on their location. And then from that's an interesting question. From my perspective, like, is VTA to SNC a gradient or not? I have no idea because we haven't even imaged at cellular resolution. And I'll say, I think it's a gradient. I have no evidence it's not. And our lateral VTA neurons, like the reviewers didn't believe it was VTA. They thought it was SNC, even though we had like clear histology that it was lateral VTA. So clearly the field thinks there's a big distinction, but our lateral VTA neurons were tuned to velocity, and they were definitely in VTA. And also, like a lot of people say, like lateral VTA doesn't project to dorsal striatum, but we see neurons that project to dorsal striatum. So I guess we haven't done anything to prove it, but as far as I can tell, lateral VTA to me looks a lot like what other people say SNC looks like. And similarly, I don't think the histology is like so clear cut that you have no VTA neurons projecting to dorsal striatum. That's just not what we've seen. So, I mean, maybe we're wrong, and I haven't like tried really hard to prove anything, but from our data, I don't see any. Our data is consistent with more of a gradient, I'd say.
Okay. Chaim, you wanted to ask a question?
Maybe a brief question about the model that you presented. Can you use the model to disentangle reward response from prediction error and history dependencies and so on?
I actually don't understand the question. Can we use the model to disentangle reward?
What the model predicts about the reward response? Is it prediction error? Is it history dependence?
By design, the model is dopamine is
but you have memory there.
Yeah, you're saying, but what we're calling the dopamine neurons is the teaching signal in the model. So by design, it looks, there's, as you said, because of the memory, actually, there's some heterogeneity. It's not 100%, like not every neuron's the same because they're calculating reward prediction error based on different inputs, but on average, the neurons respond to reward and are modulated. I didn't show this particular panel, but they're modulated based on reward expectation. So on an easy trial, when there's no surprise about the reward, there's less of a reward response than on a hard trial. So it replicates, like as you'd expect, it replicates and in like our data, like the reward response, most neurons respond to reward. Again, not everything, because there's some like noise based on this like LSTM and everything, but most neurons respond to reward maybe every neuron, but definitely some more than others, but most neurons respond to reward, if not every neuron, and then almost all of them are modulated by reward expectation consistent with RPE, which I guess, I mean, I thought you might say it's not a huge surprise. It is by design. The model is designed to try to explain what might be going on in the data. So almost by design, it's that way, but you get that. Yes, that definitely works. The model works basically.
Okay.
Yeah, okay, great. Well, thank you guys for all the questions and for inviting me to speak. Yeah, it was really nice. Thank you.
So thank you, Ilana, and see you all next week.